a biologist at Allison University. Before I get started, I want to take a minute to acknowledge the students who have done the work that I'm presenting today. Oh yeah, I was all carried out by undergraduate students. We have great students at Mount A. So most of them are pictured here. There's a few more recent ones who aren't, but in particular, I want to highlight Jada Ripley. I gave a guest lecture in the first year biology course, like 4 years ago. Jada was in that class. And a few months later, I got an email from a student saying, I saw your guest lecture. I'm really interested in trout lilies. That was the thing I was working on at the time. Are there any chances to work in your lab? So she started working after first year, and she's worked with me ever since. She just finished her undergraduate honors project. She's presented her work at national conferences. She won a prize at Science Atlantic for, I'm like, oh, I'm gushing. She's awesome. All these students are awesome. But a lot of this work is Jada's work, and Jada has decided to stay for a master's, so the story will continue with Jada in my lab. The other thing I want to say before I go too far is, I have like a presentation here, but I'm really happy to deviate from the way that it's set up. So if you have questions while I'm going or things that you want to share, please don't hesitate to stop me, and I can cut stuff out or add stuff in. We can be flexible. All right, so I am a plant evolutionary ecologist, and I'm particularly interested in interactions between plants and their pollinators. Everybody can hear me okay? Yeah. As most of you probably know, the great majority of flowering plants require pollinators to move their pollen from flower to flower to effect seed and fruit production. In other words, they need pollinators for their reproduction. And the majority of those pollinators are insects, and even more so, the majority of pollinators are bees, right? So I'm showing some diversity of different pollinators we see here. I'm showing 4 different, whoops, this thing turned off on its own. 4 different wild bees. Oh, it's okay, I have buttons. 4 different wild bee species here, some Lepidoptera. These are flower flies. They're not bees. They're little bees that look like flies and hang around on flowers, presumably looking like a fly, or looking like a bee gives them some protection, right? Other things think they might sting. Beetles, hummingbirds. But the majority of pollinators are bees. Whether we're talking about bees or other pollinators, they're not visiting flowers just to carry out reproduction for the sake of being kind, right? They're doing it for a reward. The rewards that we most often think about are pollen and nectar, and those are, for sure, the most common rewards that the flowers offer their pollinators. But there are some special flowers that offer specialized rewards for specialized visitors. Some examples include floral resins that are offered to euglossine bees. These are relatives of the orchid bees that live in the tropics. The males collect fragrances that they use to attract females. Or that was the resins. Did I say resins or fragrances first? Resins, sorry. So they collect resins for part of their nest construction. Other euglossine bees, the males will collect fragrances that they use for attracting females. But the reward system that I'm particularly interested in these days is the oil pollination system. So these are cases where instead of producing nectar, flowers produce oil as a floral reward. So these oils are collected by specialist visitors. In all cases, those are bees, and they use those oils for their own reproduction. The oil reward system wasn't recognized until 1969. It was described by a German scientist, Stefan Vogel, who published his work in German. And so it wasn't recognized broadly until the 1980s, when people started publishing in English language about it, which is a whole issue with science, right, that English tends to be picked up first. But it was first described in 1969. And again, these are systems where a plant is producing flowers that make fatty oils as a reward instead of making nectar. The females of highly specialized bee species will collect these oils and use them to provision their young. So they are collecting these oils and including them with pollen masses that their larvae then feed on. This system has evolved in at least 11 plant families, so it's occurred independently several times. There's at least 2 families of bees where this relationship has evolved. Within each of these families, both on the plant side and the bee side, there's multiple species involved. And it occurs on all continents except for Antarctica. It's most common in the tropics and the subtropics. All right, so we don't think a whole lot about oil pollination in our region, but there is one oil pollination system that does occur in the whole Arctic region, meaning Eurasia and temperate North America. And that's the relationship between Lysimachia flowers and Macropus bees. These two genera, or genuses, the plant genus and the bee genus, have been interacting with each other for an estimated 53 million years. Yeah, it's like one of the two oldest oil pollination systems. It's like dinosaurs were around, right, when this evolved. It's pretty neat. And it's assumed or thought to be an ancestral character to both the genus of bees and the genus of plants. So the very first Lysimachia plants were producing oil as a reward, and then they diversified to create the many species of Lysimachia that we have today. Same on the bee side, same with the Macropus. They were harvesting these floral oils. On the left, I'm showing one picture of a Lysimachia plant. This is Lysimachia ciliata. It's native to North America. It's not the one that I'm studying, but it's the one that's best described in North America with respect to this system. At the base of the petals and the base of the stamens being the structures that produce pollen, there are these specialized hairs called trichomes. The trichomes are glandular, and they secrete an oil like a slick, really thin film at the base of the flower. These specialized bees from genus Macropus come and visit these flowers to collect their oils. They use their forelegs, so the front legs of the bee. On the inner surface, they have modified hairs that act like sponges to mop up the oils. So this is a scanning electron microscope image of those hairs, right? They're highly specialized. Oh, bees have the coolest hairs. Like every species of bee has got specialized hairs for something in its life cycle. Bees are a whole other talk, another day. But these are the specialist hairs on Macropus bees. This bee is reaching its arms forward, its forearms forward, mopping up the oils, which will then transfer to these scopa, the hairy patches on its hind legs, where it's collecting oils and pollen to bring back to its nest. All right, so this is what's happening in this image. This is Macropus nuda, one of our 4 North American species of Macropus, and Lysimachia ciliata, which is one of about 14 native North American species of the strict definition of Lysimachia. Yeah.
So to transport the oil, they mix it with the pollen?
Yeah.
Okay.
Yeah, and then, so the Macropus bee needs these oils because most bees have well-developed glands in their abdomen called Dufour's glands that produce oils endogenously within their own body. In genus Macropus, those glands are highly reduced, and so it needs to bring these oils back to its nest. It uses them to help waterproof the walls of its underground nest. And it also uses it to mix this pollen loaf. And then it makes literally a little loaf of pollen, right? A little loaf of bread, 1 egg on top of each loaf. That's how bees construct their nests generally. But in this case, it's mixing oils into those loaves. Yeah. It's a whole other side story too. There's a different species of bee that parasitizes the nests of Macropus, and that bee's endangered, but again, a story for another day. Yeah, well, it's listed endangered in Canada. It's Epiloides silosis. It's listed endangered in Canada, but I've heard that I think the status is going to switch to data deficient. I think it's more a case of it's just so uncommon. To be a parasitic bee that specializes on a specialist bee is a pretty risky ecological strategy to take, right? And anyway, again, another story for another day. But that's our situation with Lysimachia and Macropus. Any other questions so far? It's pretty cool, right? This weird little pollination story. So the picture I'm showing you is Lysimachia ciliata. I've already told you that's not the one I focus on in my work right now. But the reason that I show that picture is because it's the one that's best understood, particularly in North America. So there are very few reports of Macropus on any North American species of Lysimachia except for ciliata. That's a statement from 1983. 30 years later, fast forward to the Kosiewicz report on that specialized parasitic bee I was just telling you about, where it stated for most species of Lysimachia, the actual contributions of floral visitors and pollination, including Macropus, have not been assessed. In other words, we really just don't know much about the pollination of these very cool plants. Like, this is cool stuff happening right in our yard, and it's just not well described. And because the focus has been on ciliata in North America, and most of the observations have been on Lysimachia ciliata, we find statements like this in the literature. I want to say I have deep respect for Brian Danforth, who's the senior scientist on this paper. Like, no, this is a statement based on the data available. Brian Danforth is a genius in the bee world. And anyway, but this paper from 2022, a very recent paper, claims that Macropus nuda, the bee we were just looking at, is only known to gather floral oils and pollen from one host plant species, Lysimachia ciliata. If that's true, what are the other North American Lysimachia doing, right? Why are they producing these floral oils if no one's coming to collect them? If Macropus nuda is only visiting ciliata, which is shown up here, what's everyone else doing? Are they even producing floral oils? That's an open question for most of our North American Lysimachia species. The ones that have an O in the corner here are ones that are reported to produce oils. So that includes the big one here, Terrestris, that I'll mostly be talking about today. The one with the X is one where we know that oil producing trait has been lost. They don't make floral trichomes anymore at all. We know that this is a labile trait that comes and goes within the genus. Or that goes, it doesn't come and go. Once it's lost, it doesn't come back. Yeah, it's pretty specialized. It'd be weird for it to evolve several times in the same genus. But this is Lysimachia thyrsifolia, which also occurs in our area. But for most of these species, I can't find any report on the oil production status. Now apparently that German scientist Stephen Vogel in 1986 published a more modern monograph for genus Lysimachia that reports on the oil production status of 170-ish species. But again, it's a German report, and it's like in a journal that I can't get easy access to. I'm working on it, and I'm hoping that there'll be a table in there that I can pull some status information for some of these plants. But the takeaway message is we don't have a good grip on what's happening in the system. And so I wanted to fill some of those information gaps, right? What's happening with the pollination of these specialized plants? Are they producing oils? If they are producing oils, is Macropus visiting? If it's not visiting, could there be selection going on to reduce the oil production trait? Right? Oils aren't cheap for a plant. Those are carbon-heavy compounds. That energy could probably be better directed to something else if no one's coming to collect the oils in the first place. Now, for the type of work I do, it makes sense to start with one species at a time. Other people approach these questions from different directions and can work with the whole genus. I'm a person who likes to go outside and see what's happening, right? I want to just go and watch a plant. And the plant I started with is Lysimachia terrestris, or swamp candles is the common name. Does anyone recognize it? Yep. Yeah. So this plant grows all around Sackville, which makes it a great study system for work in a university-centered research facility, right? Or an undergraduate-centered university. It means I don't have to send undergraduate students far away to the tropics without me and hope that they come back safe and they can manage. They're literally in walking distance of their field sites working here in Sackville. I first saw this plant in my backyard. I live near Silver Lake a few years after I moved to Sackville. I was like, oh, what's that? And I looked it up, I figured out what it was. I read that it's part of this genus of oil producers, and that's it. Like there was nothing else. It just seemed like a massive opportunity to start investigating what's happening. One of the things that intrigues me about Lysimachia terrestris is that its known range, which is shown here on the right, I know this is not a great figure, it's hard to see, but it goes all the way up into northern Quebec, wow, into Newfoundland and Labrador, right? It far exceeds the known range of that specialist bee, Macropus, which is limited in Canada to pretty close to the US border. So I think it offers some interesting opportunity to ask what happens to an oil pollinating plant when it's beyond the known range. And when I started working on this system, there were no reports of the Macropus bee from this part of the Maritimes. There were some reports from further south in New Brunswick, and some from Nova Scotia, from Corey Sheffield, who some of you may know, maybe not. But there was nothing from this Tantramar region. These are observations that were added by my lab. So, spoiler alert, we have found Macropus right here in town, and I'll tell you more about that in a minute. So yeah, one of the questions I want to get a grip on is what's happening with Lysimachia in these other areas. Not necessarily like far north Quebec, that seems a hard place for me to do work, but Newfoundland's accessible. And I'm also curious about what happens where Lysimachia terrestris and Lysimachia ciliata populations are growing close to one another. Ciliata is that species that's reported to be the preferred host of Macropus nuda. So do we see them competing with one another? Does Macropus really prefer ciliata over terrestris? Right? There's some cool biogeography questions that can be asked here. I've already summarized those, so I'm not going to repeat this slide. And so I'll jump straight into telling you some of what we found. Questions so far?
Yeah, if you have swamp candles, you have to be.
We do here. We do here. Do we have them everywhere? Not necessarily.
So, there's a lot on my property.
You have a lot of swamp candles?
Yeah.
I would love to come look for the bee one there.
You're welcome.
Yeah, awesome. Because, well, so where we have been doing our work so far has been the studies, the retention pond that was newly constructed at Lawrence Street and St. James Street. And what I'm observing there is as it gets more and more vegetated, the swamp candle population is dying back a little bit. I think it's a plant that likes recently disturbed areas. At least that's my observation at this site. So it's been a good study site for about 5 years. And I think some of the newer parts of the retention pond, we can start moving into those as the population at the originally constructed site at St. James and Lorne starts to get more and more vegetated. But eventually I'll be looking for other places. Yeah. But yeah, we have been working at this retention pond. You know it pretty well, right? Everybody's familiar with this area. Yeah, so I won't go into too much detail. My understanding from Jamie Burke is that when this site was constructed, they collected the soil. Some of it had to be remediated, but not all of it. Some of you were more involved in town affairs probably at that time than I was. Because I think it was a big deal in town when it was just, yeah. Anyway, the soil was collected and then returned to the site. So the vegetation that grows there is largely from propagules, plant propagules that were in the soil, right? It's sort of a naturally colonized area. This is what it looked like after construction in 2019. And if any of you have walked there recently, you know it's like the vegetation is much denser now. But I found swamp candles growing there in 2020. And that was a really convenient study site. In order to get a grip on this system, we had to answer a few basic questions. One thing I needed to know was whether the flowers were self-compatible. Those of you who study plants probably know what that, or work with plants, know what that means. But essentially what it's saying is, does pollen from the same plant work to produce fruit on that plant? Many plants have incompatibility mechanisms that mean that it requires pollen from another individual of the same species to affect fruit production, right? And so I needed to know whether or not swamp candles were self-compatible at our study site. Again, very little information available, so basic question. I'm not going to go into the details, but essentially we carried out hand pollinations using pollen from known sources to pollinate flowers. And from 2 different experiments, we found good evidence that they are self-incompatible, which means they can't produce fruit on their own, right? They can't just pollinate within a flower. Instead, they need some animal vector to move pollen from one flower to another. So the plants need animal pollinators. Are they setting fruit in Sackville? The answer was yes. Not a lot, but yes. So these are bars representing 74 different plants in this population in 2022. The height of the bar tells you how many flowers that plant had in that study year. And the yellow part of the bar is how many fruit it made. So there's not a ton of fruit being made, but there are fruit, right? It's not zero. And some of the plants are doing pretty well. This one's got an over 75% fruit production, but it's not super high, you're right. They are also highly clonal plants, so that could be part of what's happening. But it's not zero. So something is moving pollen around this site. 30 out of the 74 plants, which is about 40%, made some fruit.
What's the fruit look like?
They look like, I have a picture somewhere in here. They're not flashy fruit. Let me see. I have so many extra slides.
Yeah, I don't think I've ever seen it.
So this is a picture. I tried to do some, I'm not going to tell you about this stuff today. So many stuff that I ended up cutting. This presentation would go way too long otherwise. This is an image from iNaturalists. We've been trying to do some looking at iNat photos to see, can we see where in the range fruit are and are not being made. This is a fruit.
Okay.
That's a fruit. So it's a small dry capsule that forms a sphere. And on the other hand, these are flowers that did not mature into fruit. All the ones where you can still see the sepals and it hasn't swollen. Yeah. Right, so some pollination is happening. Who's pollinating these plants? The first year we looked for it, we found Macropus, right? We didn't see a ton of it in 2021, but we saw some. This picture is actually from 2024, but we observed some bees in 2021, which I think is remarkable because remember what that site looked like in 2019.
Right.
So that's a really clear indication that there are Macropus populations around here, speaking to the question you raised, that were ready to recolonize that site as soon as it was available, right? It's not like a long-standing population. Those are bees that moved in after that big construction project took place. But Macropus wasn't the only thing visiting the Lysimachia.
Oh.
We also saw a lot of Lasioglossum or sweat bees visiting. These are different species of sweat bees pictured here. Again, all these pictures are by Jada. She's so good. She's so good. She can like take notes on a voice recorder and photograph and mark flowers all at the same time somehow. She's really good at what she does. But these are different sweat bees that are visiting Lysimachia flowers at a retention pond site. You probably, like, you see little flying black bees that land and lick your sweat in the summertime. That's these friends. And we also see a lot of visitation from flower flies, the syrphids. Both the sweat bees and the syrphids are expected to have zero interest whatsoever in the oils. They're there for pollen only. And to the best of our knowledge, they're generalists. They'll take pollen from Lysimachia, but they'll take it elsewhere too. The other interesting thing we found is that the specialist Macropus is pretty uncommon relative to those other generalist pollinators. So these are data from 2023, these are 2024. And we're looking at how many of each taxon did we observe. Generalist bees, generalist flies, the specialist Macropus. U just means unknown. We couldn't discern what that was confidently on the voice recording. In both years, the Macropus are pretty uncommon relative to the generalists. So then why is the plant bothering to make the oils, right? If Macropus is hardly ever coming, why not just rely on these generalists instead? Well, maybe Macropus is uncommon, but is still more important. And that could arise if its frequency is low, but its intensity of visitation is high, so when it does come, it interacts with more flowers, and or if it's more effective on those flowers that it does interact with. Is it more likely to produce a fruit? So we've set out to answer that question. We find indeed that Macropus is more intense a pollinator. It's a weird phrase. I don't even know if it's the right word for it. But when it comes, it tends to visit more flowers per visit than do bees or flies. So within a 2-minute interval, when a Macropus arrives, it visits on average 11 flowers in the patch. Whereas bees and flies are more like 4 or 3 flowers within a 2-minute interval. And a cool thing, I was talking to Jeff earlier, I'm on sabbatical this year, which means that I can read and think about like my work in a broader context in a way I haven't been able to in so long. So I've been reading about a bunch of other oil pollination systems. Not surprisingly, most of the work on oil pollination is from the tropics because that's where it's most common. And it is really common for oil bees to visit flowers very quickly, like spend a second at most on a flower. And that's what we see with the Macropus. It visits more flowers per unit time because it lands on it and it moves on right away. It doesn't spend much time on the flower. And that seems to be something that happens with oil pollination. Okay.
So it's hard to get a photo of it.
Yeah, exactly. So Jada's photo is all the better. Exactly. Yes, thank you, Jeff. So it's more intense. Is it more or less effective than the generalists? To answer that question, we have to observe a bee approaching a patch of Lysimachia. This is a Lasioglossum, it's not a Macropus. Macropus are hard to photograph. It's flying up to a patch of Lasioglossum, of Lysimachia. We watch which individual flowers it interacts with. Yeah. After it visits 2 flowers, we chase it away because you can't keep track of more than 2 flowers at a time. We then mark those individual flowers to say we saw a bee or a fly of this species interact with this flower. We also marked 2 flowers on which we did not see visitation to be sort of a background pollination data. And then we bag the plant so nobody else visits. A few weeks later, we return to see whether or not it's setting fruit. This one is, and we count the number of seeds inside the fruit. What we found through this experiment, I did this project, Jada did this project in 2023 and 2024. In 2023, there weren't as many observations in 2023 anyway because we were learning the methods, but too many of these bag plants were near the edge of the path at the retention pond.
Oh.
And about a week before it was time to collect the fruit, the town mower, we have site permission to work there, but that's not necessarily communicated to the teenager who mows. And so like half the experiment was mowed down. Oh yeah. But an important lesson, right, for everybody in the lab to learn, take at least 2 big steps away from the path before you mark a plant. Have any of you noticed these bags around the retention pond in past years? Yeah, we always clean them up. So they look like a bit of a mess, but this is what we're learning from them. But across those 2 years, we have found that Macropus is indeed a more effective pollinator. So these are the 3 taxa, the specialist bee, generalist bee and the fly. Micropus, out of X number of flowers visited, 65% of them mature into fruit. The number is much lower for bees at 25% or 30-ish% for flies. So it's a more effective pollinator. The other weird thing going on with Micropus is that the flowers on which we did not directly observe visitation are also more likely to set fruit. I have some ideas about why that is, that if you want to know, I'm happy to chat, but yeah.
So, I have a question.
Yeah.
So, you come to the plant, you don't know who's been there before.
Yeah.
Okay.
So that's what I think it is. So ideally, an experiment like this, you would bag the plant ahead of time,
Yeah.
and then you're visiting flowers that are not previously visited. But when you're dealing with something as rare as Micropus, where we see on average 1 per 3 hours of observation, yeah, we can't rely on that method. So we're doing this as a proxy instead. But what I think is happening is that the Micropus tend to be patchy in their visitation. So if they're showing up in one spot today, there's a bunch of them showing up in that spot, right? So there's a good chance that a flower on this plant was previously visited by Micropus. Like, that's my gut. It's hard to prove, but that's what I think is happening. Yeah. Flowers visited by Micropus also set more seed than do flowers visited by bees or flies, which is cool. So they are definitely more effective, and I think it might have something to do with how quickly they move through the flowers. They might be depositing less self-pollen within the flower, and we know that pollen doesn't work on these flowers. And it also has to do with how they handle the flowers, I think. The generalists are only interested in the pollen, and they just dance up around on their tippy toes on the anthers and the stigmas. So we see the bee here just kind of circling. The fly, Jada describes it as they like to bop. They just bop their abdomen and eat some pollen. Whereas Micropus gets right in there, right? It uses that same posture we saw in Ciliata. It's holding the top of the flowers with its hind legs and plunging its forearms right in. Its abdomen makes really good contact with the stamen and the stamens and the stigma in that posture. So there's some behavioral differences. When we think back to importance and we put it all together, we find that Micropus seems to be more or roughly the same or potentially even a little bit more important a pollinator in terms of number of seeds produced for 20 minutes than the flies or the bees. So there, again, our importance calculation, we don't have to worry about, these are numbers that I showed you before, you don't have to worry about the depth of it. Essentially, it's just a multiplication function, and it's expressed in units of number of seeds per 20 minutes of visitation. The flies, we expect about 2 seeds per 20 minutes. The bees, about 4 seeds, and Micropus, about 4.5 seeds. Now, this looks higher than this, but in reality, there's error around all these estimates, right? And it's hard to propagate that error through these calculations. There's also assumptions in here. So my conclusion is that they're about the same. Micropus and the generalist bees are comparable. So again, it's raising the question, should this plant continue investing in oils, or would it be better off saving that energy and maybe investing it towards maturing more of the fruit from lasioblossom visits? I don't know. In the interest of time, I'm going to skip the next 2.
But if it's been doing it for millions and millions of years, it can't change its mind, can
Oh,
it?
why not, right? That's how evolution works. So these 2, sorry, Ted had a question too. It might relate to the thing about biogeography.
So I'm just curious whether the oil is involved in the pollinating process.
That's a great question, yeah.
Because
So,
that would perpetuate that was needed.
right, so maybe getting the oil on the stigma somehow helps the pollen do its job. And yeah, good question, and it was, to my knowledge, not explored in oil pollination generally. Some of the reading I've been doing this week suggests that somehow the process of oil pollination and the posture that oil bees take, not in this system, but in other oil pollinated systems, helps to remove some membranous layer from on top of the stigma. So I'm going to look at some of our preserved flowers and see if I can find evidence of that. But yeah, whether or not mixing the oil in with the pollen makes a difference, that sounds like a great undergrad project. Yeah. And then the other, so to Harold's point, these have been interacting for millions of years. How could it be that that trait is lost in some populations? Am I doing okay? Yeah? Am I on time, Jeff?
I don't have a watch. I'm
Okay,
dangerous.
it's 40. Should I stop here?
We're entertained.
Well, people have
Wait,
places to
you
go.
can start to
Wind it
land.
down?
Start
Yeah,
to.
so I have
And you might have some
biogeography ideas
questions.
for another day. But what I do wanna, the last thing I'll tell you about, so this is a slide I showed you earlier. I told you something about what's happening in Sackville. Tantramar is just one place, right? I don't know how this system is behaving elsewhere in its range, and that's a question we're trying to get at. What's happening in these areas where Macropus has never been reported? So this summer, Jada, who is staying for the master's, this is the beginning of her master's research, brought other researchers from my lab, Jasmine and Hannah, the three of them went on a 4-week odyssey to Newfoundland to see what they could see, right? Could they find Macropus visiting? They started in Port aux Basques, drove across the island all the way down to the Avalon Peninsula, spent some time there. Eventually sites got closed by the horrible wildfires we're experiencing. But the plan always was for them to return to Port aux Basques and stop at populations again on the way back, catch the ferry home. Across the 4 weeks, they visited 30 sites. They found Loosestrife at 14, and they did 71 hours of pollinator observations. I only have very preliminary reports here because they only returned, mid-August. But some things we find, one, there's a lower probability of anything visiting Loosestrife in Newfoundland than there is in Tantramar, at least. In Tantramar region, for a 20-minute observation period, we would see something visit Loosestrife with a 50-50 probability. Something would come 50% of the time. Not necessarily Macropus, but something. In Newfoundland, that value was 25% probability something would come. So about half the visitation rate for just any visitor at all. A second finding, Macropus was absent, or very rare, right? I love your gasp, Harold. It's like the best movie, right? Yeah, there was no Macropus, and there have never been Macropus reported in Newfoundland, but there's never been a concerted look for it the way that my lab has carried out. So this is sort of corroborating the idea that Macropus either isn't there or it's pretty rare. For the number of hours that they observed, if there were as many Macropus in Newfoundland generally as there are in the Tantramar region, we would have expected to see something in the mid-20s, right? And we saw 0. They saw 0. I didn't get to go on this trip. The other thing we see is that there's a reduced fruit production in Newfoundland relative to Tantramar. Now, they were there during flowering, so it's a little harder to score fruit production, but on 39 of the plants that they saw across 8 study sites, they could score whether or not fruit were forming. Only 1 out of 39 plants had any sign of fruit forming.
Wow.
And compare that to 30 out of 74 in Tantramar, right? So there seems to be lower fruit production. So everything to me is indicating that these sites are different. And I think that's really interesting, and I'd like to expand the work further and start seeing what's happening with introduced populations of Loosestrife terrestris on the west coast. So this swamp candles is an invasive weed, it's a weed in BC and Washington State in cranberry bogs. There are Macropus bees in BC, but we have no idea if they're visiting and helping to propagate this plant and sustain these populations. But more generally, I think that looking at how pollinator communities differ in different parts of a range, and then going further and asking how do plants respond to those differences in pollinator communities? Do we see an evolutionary loss of the oil reward? Do we see other evolved floral traits when pollinator communities differ? I think these sorts of questions can give us a window into the plant responses and the ultimate effects of contemporary change in pollinator communities. Many of you are probably aware that pollinator service is declining globally. I was doing some quick Googling before we started, and the earliest report I can see claiming a global decline in pollination is from 2003, so this is a long-standing trend of declining pollinator service. Those declining pollinator services and declining pollination isn't affecting all taxa at the same rate. So while we see declining services, we're also seeing changing composition of pollinator communities, right, which can also affect plant populations. There may be more generalists and fewer specialists, for instance, right? We expect plants to evolve in response to changes to their pollinator community. The study of plants and their pollinators and how they evolutionarily interact with one another is one of the best examples we have of coevolution. We expect to be plant evolutionary responses, and a reasonable response to a plant with a specialist pollinator is to evolve away from being specialized for that pollinator if the pollinator's not there. Like a reduced oil production, for instance. But that in turn could create a negative feedback loop and further harm a pollinator and community in decline. So if we're interested in pollinator dynamics in a changing world, I think we need to think not only about the threats to pollinators, direct threats, and how to mitigate them, things like pesticides and global change and urbanization, et cetera, but also about how plants evolve in response to those threats and how those evolved responses in turn go on to further affect pollinator communities. There's a an important feedback loop that's taking place there potentially. And I think Loosemachia, with its geographically variable Macropus visitation, could be a model for these types of responses, using space as a proxy for time. So that's kind of the bigger picture. Okay.
Can you measure the amount of oil? Like, can you
Yeah.
can you show whether there's more or less oil?
It's something I really want to do. I've been reading on methods for that. One thing I have tried is counting the number of trichomes, so those hairs per unit area, with the assumption, One thing I've had undergraduates do is count the number of trichomes per unit area under a dissecting scope. And that's one way to get at it, but there are other methods where the oil is a really thin film on the flower, so it's hard, it's not like you can collect a volume of it. But with nectar studies, where nectar is a thin film, some people use biochemical methods where they wash the flower with a known volume of solvent, and then you can use things like
Concentration.
chemical methods to measure the oils within it. I wouldn't be able to use water as the solvent, but there are other things that oil probably dissolves into. I need to talk to a biochemist. My husband is a biochemist, so maybe, yeah, he can probably help me figure that out. I'm gonna show you one cool picture, and then I'm gonna give my takeaways, I promise. We've also been playing with SEM imaging of these flowers. This is what the trichomes normally look like under SEM imaging, right? But in our study population here, we occasionally see plants that produce trichomes that look like this, that are all wonky, which maybe they're not functioning properly. There's something developmentally wrong with these trichomes. So that's kind of another area we'd like to go. Find some of these clones that are making weird looking trichomes and find out if they are actually making oil or not. And is this morphology more common in Newfoundland than it is here? I don't know. Okay, so lots of questions. The main takeaways, number one, absence of evidence does not equal evidence of absence. This is how it was phrased by Zach Brown, a former undergrad of mine. When I started this work, I didn't think Macropus was here because it hadn't been previously reported. I thought, how cool, I'm just north of the known Macropus range, this will be great. And then as soon as we start looking, we see it, right? So in the case of Tantramar, it was a case of absence of evidence. In the case of Newfoundland, I think it might actually be evidence of absence. I can't say definitively that it's not there, but we tried and it wasn't, right? So we're hinting more towards true absence in Newfoundland. Another takeaway, there's all kinds of fascinating natural history in our hometowns and our own backyards. You already know that, but sometimes I really drive that home to an audience. A third takeaway from a pollination perspective, the most common pollinator is not necessarily the best pollinator for a plant, right? We see that with Loosemachia. The uncommon Macropus bee does a better job. And then the fourth idea, that this cool system we have in our region in the Maritimes could be a good model for thinking about how plant populations will respond to changing pollinator service over time. So many students have participated in the project. So many colleagues, both at university, or Mount Allison University and beyond. Site permissions for all of the work, always, including in Newfoundland, of course. Several funding agencies have supported the work. And I gained a lot from local knowledge within the town, too. Yeah, thank you for your time. And I hope I didn't go too long. I'd be happy to take questions.
Other questions? There must be a few.
Or just keep an eye out for the plant. And so it grows in my backyard. I have one patch of it that makes a ton of fruit, and I wonder if that's a patch that's lost its self-incompatibility and can self-fertilize, or maybe there's Macropus in my backyard, but I just haven't spent enough time looking for it.
Really?
Mm-hmm.
So how would I learn about the different bees and what if I,
It's hard.
it's hard?
Yeah, so of the ones that visit the Loosemachia, the Macropus it's a really primitive looking bee. You'll
But
see
do I have to take a picture of it and?
if you can get a picture, you can send it. You can also, the easiest way to photograph a bee is to capture it in like a pill vial. It's not that hard. Macropus is tricky because it moves so fast. But capture in a pill vial, put it in a cooler with ice packs for 2-3 minutes, or in your fridge for 2 or 3 minutes. It'll kind of take a cold weather nap. You can then put it out, take a few photographs, and it will warm up in the sun and fly away. Don't freeze it, that would be over, and don't leave it there indefinitely, but for a few minutes, that would slow it down. You can take a picture easily. Or you can try photographing on the flower. Jada's had great success with just her iPhone, right? I don't think most
Really?
an iPhone. Those are all just cell phone pictures. that she's-
Seriously?
Oh, yeah.
Not
Yeah,
bad.
she's good. Yeah. Yeah, she's really good. And then she crops them, right,
Yeah,
to
yeah.
like bring them into focus, but yeah. Yeah,
But they're still pretty sharp.
they're very good, yeah.
Yeah.
She takes, for every one that's here though, there's
Of
probably, 60 that aren't,
Yeah.
but yeah, she does a good job. She does a very nice job.
Well, my question connects with Harold's in, can you describe how to identify one of these particular bees to us, or is that?
Yeah, so if I
pretty
was
hard.
if I was looking for Macropus, there's two places I would look for it. One is on the swamp Campbells. The second is on the dogbane, Apocynum.
Okay.
Dogbane, we have some growing around the retention pond too. What dogbane? There's spreading dogbane, I think
Yeah,
is the
spreading dogbane.
common name for it. Macropus love collecting their nectar from that plant for some reason. I don't know why.
Is it oil? Is
No,
it-
it's getting nectar.
Oh, it's nectar.
So they need nectar too. They just can't get nectar from Lismachia.
Oh, my goodness.
But they love so many reports describe them nectaring on dogbane, including there was Macropus nuda described in Washington State for the first time in I want to say 50 years, just I think within this past year there was a report of it, and it was observed on dogbane, not on Lismachia. So they really like that plant. That's a good place to watch for them. But if you're watching Lismachia and you want to know if the pollinator visiting is Macropus, I would look for something that's moving very quickly through the plant, and that it's pitching forward. I would watch the behaviors more than I'd watch for the bee itself. They're also more robust than the Lasioglossum. They're slightly larger. And the Lasioglossum are kind of a finer bee. Like I say, Macropus is part of the Melididae, which is the first family of bees to differentiate from the rest of the bees. And they just still kind of look primitive, for lack of a better word to me. Yeah.
Hmm.
Yeah. But pay attention to bees. Bees are
Well,
Oh, yeah.
and if you get photographs, you could submit them to our naturalists. and
Absolutely.
see.
Yeah.
Yeah. So most bees you can't identify to species from a photograph.
Okay.
You can probably get to genus for a lot of them. Certainly usually to family. I have a whole other separate project that some of you may have known about, where we were documenting bee diversity in the Tantramar region, in people's backyards and at the wetlands
Oh,
around
yeah.
town. I'll be
Yeah.
talking a little bit about that on Thursday at the quarry opening, just like a 2-minute spiel. We're still
The
working
people
on
had
this
those
yet.
boxes?
Yeah, we
placed
had,
in-
yeah, I kind of have moved away from bee hotels as a thing
Oh.
that I want to promote for reasons I can get to. I just don't think that habitat is the thing that's limiting native bees in Tantramar region, and I think just this whole other subject too. I think that there are some ways that we can, leave some standing vegetation, leave some bare soil patches for ground nesting bees. I think that's probably the best thing we can do. Most of our native bees are ground nesters anyway. They don't use the boxes. Another talk for another day. But yeah, we have been working on documenting bee diversity in our town. I have a nice reference collection of bees now that are identified to genus, and we're working on getting them to species. It's hard to get bees to species.
Oh, boy.
It's hard. Some you can do, bumblebees you can do from a photo pretty reliably, but a lot of them are tricky. But you can look at it and say that's a Lasioglossum or that's a Macropus if you're looking at visitation on a Lismachia plant. Yeah. And sometimes that ecological context helps, right? What plant is it on? What time of year is it? Yeah. Ted, did you have a?
Yeah, I was wondering, can you propagate Macropus in captivity, and if so, does that lead you to opportunities to have controlled
Yeah.
testing of this story?
I don't think so, is my short answer.
Is it unethical to take them
I have,
for
so
their cycle?
when sometimes people are able to keep bees indoors. I've seen reports, so I know bumblebees for sure, right? Because they're moved around in boxes for pollination anyway. Honeybees, obviously. Bumblebees, certain types of bumblebees, yes, others no. Bombus impatiens, it works. Bombus Trinarius from the UK, they'll put in boxes and move around for greenhouse pollination. But other native bumblebees, no, right? And then the Macropus has so many specific needs. It needs its oil host, it needs a nectar source. It's only active for a few weeks of the year, right? The adults are out for during Lismachia flowering, you won't see them anymore, they die. They emerge as adults, they do their life cycle, they build a new nest, everybody dies, and they overwinter as larvae. So providing all of those biotic and abiotic requirements, I just, no, I don't think it's something I could do. What I can do though, and what I'm working towards is rearing Lysimachia, the plant, indoors, and so I can bring plants out for tests. So one thing I'd like to do,
You mean in pots?
yeah,
Huh.
yeah,
I
is
can give
get,
you pots.
sure, but get Lysimachia from Newfoundland, where
Oh, yeah.
Macropus is uncommon. If I see that there are differences in the flowers in Newfoundland versus here, bring some potted Lysimachia, put it outside at the site where I know that Macropus visits, and see, do they prefer the local Lysimachia to Newfoundland Lysimachia? Like, do we see that there's some effect on pollinator service because of the way those plants have evolved in Newfoundland? So that's feasible, but trying to keep the bees indoors. Nope. Yeah. Yeah. Thank you for your interest. I appreciate it. If anybody has other questions or you just want to talk about bees or plants, that's my email address. Yeah.
You're good.
Thank you.
Lucy's gonna thank you on our behalf.
Oh.